So the first thing I would like to say on a lighter note is that COVID-19 yes. Yep. So those were the days where we used to go socialize with people, interact, and visit so many places. And as soon as this pandemic has hit, the whole world has gone so empty that we are not even able to speak to others with wholehearted nature. And then we had to suspect everybody as if they got coronavirus or something. So this is what we are in now, and what this pandemic has done. And literally, in a short sense, the whole COVID-19 has shattered the world. So, ma'am, am I audible, ma'am? Hello?
Yes, sir. Yes,
okay.
yes.
Thank you very much. Yeah. So, from a free world, we have come to a position where we have to wear masks all the time as we go out. It's really a punishment for us, and this is what you see all the time. And here I am to present to you what we don't see in the lab to keep everybody safe and sound. So these are the things that you don't see normally in a day-to-day life, and which is playing a greater part of a role in terms of disease, in terms of containing the disease as well as preventing it from further progression. So this is a PCR CT graph from one of my runs. So in this presentation, what I would like to present is one part of my work that I'm doing at Genesis Diagnostics since February the 14th. So in this presentation, I would like to present you the molecular diagnostics or molecular diagnosis of COVID-19 and why testing is the key. I know you have so many people talking about we need to test, but why is it so key? Because I don't think nobody has touched on this part. And COVID-19 Genome Assembly, and this is a major part in terms of designing the diagnostic kit and even the NGS, and what kind of a role this plays, and what kind of discrepancies that we're going to get will be discussed. The factors in designing the COVID-19 diagnosis test, and it's not only pertaining to COVID-19, but it applies to all the diagnostic tests. And we also did, we are not only a diagnostic lab, we are also a hospital as well. So we have a 120-bed hospital, and we have the finest doctors in the world, mainly pulmonologists. So I would like to share a small study that we have conducted out of curiosity to see how this COVID-19 diagnostic kit that we designed is working. So coming to the first thing, which is COVID-19 diagnostics. So detection is the key to contain COVID-19, because the first thing that anybody having a little bit of symptom, it doesn't matter whether it is related to COVID or some other flu, is to go for a diagnostic test. So why is diagnostic test key? Because this gives us an exact position of how the patient is, and in terms of his to design his treatment strategy on anything. So with respect to COVID-19, as of now, there is treatment. It's only passive treatment that's going on. So detection is the key for COVID-19, and detection after getting the symptoms and before getting the symptoms, which makes a lot of difference, because it's as everybody knows that the COVID-19 is a very rapidly progressing and with a high reproducibility rate of 4.5. So early detection of COVID-19 is a very important or a late limiting step in terms of cutting down the further transmission of the virus. So I would like to present this with a small graph actually. So this is what happened to us in India. So if you see this small curve there at the very bottom, which is in a dotted dashed line. So this is where we are at in the month of May or end of April. like that after the introduction of the lockdown. So, and if you look at the very high bell-shaped curve, which is a spike, these are the number of cases that you will be seeing. This is a classical example in the US, basically. And I'm talking about New York, Pennsylvania, as well as Texas states. So if ever there is no lockdown, this is the number of cases that you're going to see with respect to the spike. And India, being very cautious about it, has introduced the lockdown, but unfortunately, due to some, or we have to move our economy going, so we have to take off the lockdown. So we will be reaching this bell curve in very shortly, and this is inevitable until we will find a concrete view. So at this juncture, why is detection important? So detection is very important to make this whole bell-shaped curve a very flat curve, like the ones you see on the at the bottom. So as soon as you see a person positive for COVID-19, he has to quarantine because there are a lot of factors that influence it. So which I'll be sharing in my next few slides, which we have done a small study. So quarantining a patient who is positive, despite showing no symptoms, is a key step in preventing or flattening this bell-shaped curve. And this is the aim in terms of and also slowing down the community transmission of COVID-19. So I think this is a slide that you might be seeing a lot of times. I don't want to go deep into this. So COVID-19 is one of the SARS family of virus, which shares all about 85 to 86% of genomic similarity. Despite that, but it has a lot of reproductive capability compared to SARS-CoV-1. It's a 30 kb genome, which is compared to other genomes, it's pretty much small. And the key for designing any diagnostic test lies in selecting which gene or which gene you're targeting. So this is one thing that we need to take home from this whole genomic assembly of COVID-19. So coming to the next part, which is the slight symbolic way of representing the amino acids of spike protein and non-structural proteins on COVID-19 genome. So if you look at the initial kits that were designed by major institutes like CDC, Center for Disease Prevention Control in the US, as well as World Health Organization, they have come up with a very strange genes. Like CDC is only concentrating on nuclear protein genes, as well. If you look at the WHO, they look at RBRP and E gene. So these are the two recommendations by the world famous institutes for containing all the pandemics, and who are doing it very closely in terms of its transmission. So we haven't, as of our company goes, we did not take any of this into consideration. We have designed our own kit and validated it in our own lab and submitted to the FDA for emergency use authorization. And we got the authorization in the month of May 25th. So, and I would like to share some of the details of our validation as well in terms of designing it. So if ever you want to use any kind of a COVID-19 kit to design, or look at the primer designs from CDC, you're free to go to the website of CDC or WHO to find the primer sequences. There are numerous first primer sequences, and the numerous sequences from the day one. So what are the main factors in terms of designing the COVID-19 diagnostic test? So the main factors are nothing but the primer design itself. So we took RNA-dependent RNA polymerase as our target gene for designing the primers. So, and what we observed during our experience with COVID-19 diagnostic test is the quality of the swab is is a crucial factor in determining or limiting the false positives to false negatives. And this is a key step. And most of the people think that I have given a swab, and if ever the healthcare worker or a nurse, whoever is responsible for taking the swab, is not swabbing well, really impacts the way the results come out from the COVID-19 diagnostic. So it mainly applies to molecular diagnostic tests, especially RT-PCR and NGS or LAMP test. So coming to the next point, which is the swab collection. So the spot of swab collection is a very important step, because normally people think that, okay, it's the same, it's a systemic infection to the upper and lower respiratory tracts. But it does really make a lot of difference, which I'll be sharing you some of the data that we have done in our lab. So the spot of sample collection makes a lot of difference in terms of getting a positive and a concrete positive and a false positive results. And finally, this is the stuff that we don't get normally used to in our everyday life, which is specificity and sensitivity. So these are the two crucial factors, or two sides of a coin for any diagnostic test. So COVID-19 test has to be designed in such a way that we need to limit the number of false negatives as opposed to false positives. Even though if a person is false positive, it doesn't really make any big changes to his life, because he doesn't get any symptoms of that. But if it's a false negative, he would infect so many people down the line. So specificity and sensitivity are biggest bottlenecks in terms of designing a COVID-19 or any diagnostic test. So we need to kind of go to the rationale where we need to optimize these two parameters such that they're in imbalance. So it's like moving the center of mass of a car when you're turning around a steep curve. And coming to my first so this is the graph that we got from one of my technicians, where we have collected almost 150 swabs from different people, and out of that we selected 41 patients who are very positive. I would say, like we gave them triple positive. We had an internal rating. So as you could see that the throat swabs and nasal swabs have a completely different way of giving us the results in the early days of onset of the symptoms, which is a very crucial factor. As you could see that there is the black line, which is the throat swab, has in the crucial period, which is called the window period of infection, the viral load seems to be very low, as you can see from the CT values on the y-axis. And the CT values from the nasal swabs correlate with the exact positive disease state of the patient. So it is this reason that we insist on having a nasal swab rather to a throat swab or an oral swab. So this is one of the findings that we kind of did internally to make sure that whatever we are swabbing is the right spot, and which is a key designing factor in terms of the COVID-19 diagnostic test. So and one of the factors that I would like to discuss out of all our COVID-19 testing results is that there were high viral loads that were detected as soon as the symptoms were onset. And molecular diagnostic, or maybe the RT-PCR, played a very crucial role in terms of determining positiveness at the very early stage of infection before the onset of symptoms, as you can clearly see here for the 41 patient data that we have. And one interesting thing that we kind of noticed is that some of the patients that we found positive do not have any symptoms, and while some of them had, which correlates with the age or a high risk group, or more than 55 age group. It also depends on the BMI. And what we found was even the asymptomatic patients had a viral load similar to the symptomatic patients, but they do not have any symptoms, but they've been transmitting this virus to so many people since the last 4 or 5 days since their infection. So we are very intrigued by this discovery, actually, in terms of being asymptomatic and transmitting to so many people. And this is the reason why it is so crucial to test the patient or a person who is in contact, maybe a primary contact or a secondary or a tertiary contact. So it's a very crucial factor, because he becomes a super spreader, or he or she becomes a super spreader. So this is the reason why it's very, very important. So coming to the next factor, which is the specificity and the sensitivity. One of the very important thing when designing a diagnostic test is that we need to understand the cross reaction or analytical specificity of the test. So as coronavirus being a very close relative for coronavirus family, so it's always important to determine whether we are testing the right COVID-19 or not, or SARS-CoV-2. So we need to test a list of compatible, closely related viruses, and which is a key step in terms of designing a diagnostic test. So this has to be borne in mind, and so whatever test that the people design, they have to look at the close relatives, and there are very good machine learning programs to do this online. Some random forest models that you can go for. There's a lot of coding that can be done on this respect to kind of find the right parameter set for COVID-19 test. So if you have any kind of interest on this part of the area, I would definitely help anybody who's interested in terms of designing it. And we have an in-house program, parameter sets. And coming to the last part, which is specificity and sensitivity, while we are designing our kit, is that what we have tested is we have tested some patients where but a very good percentage of positives and with a negligible number of false positives and false negatives, basically. But in reality, whatever samples that we did were not on real samples. So the samples that we tested for emergency use authorization were all contrived synthetic samples. So I don't think this really implicates the real-time scenario. So whenever designing a diagnostic test, it's always good to go with the real samples or real-time samples. This is one of the major factors that really influence. And looking at this data, anybody can say that this diagnostic test is the best one. In reality, it's not like that. It's a completely different story, which I'll be sharing with you in my next preceding slides. So as I told you that I'm not convinced with the way the contrived sample study went through and validation went through. So what we have done is we have at our disposal Jefferson Hospital, which is our main partner. So we have done a small study with people of 41, and this is the data that we had from them. So this is one of the that we have taken from the patients through nostril swabs, and we limited ourselves to a cutoff of 25, an average CQ value, and selected 41 subjects for the study. And out of this 41 patients, we had 55 of them. 8 of them greater than 55 years old, and the rest of them are below 40 years. years old. Just to give you like a small demographic. The reason why we took 25 is because whatever study that we did were consistent with this value of 25 being more positive or a triple positive on our scale. So this is the determining factor, and also we did some ROC curve analysis to find out this average CQ value. And if you're interested, I can share the data with you afterwards. This will be published in next coming months. So this is a small diagram that one of my colleagues put forward actually with the 41 patients. So this gives a clear indication of how the whole diagnostic test of ours correlated with the exact matching of the clinical symptoms. So on the 7th day of these 41 patients, these people, they came to our facility at different dates. Hence, we did not give the exact date of admission, and we took this as a random 4-day. And then these people were admitted into our hospital on the 7th day, which is 1 week after the test is done. On the 8th day, we found that 51% of the patient had a shortness in breath. And so some of them had very serious symptoms, respiratory distress syndrome. And 16 of them, out of them, they succumbed to death to COVID-19. And sorry, it's 6 of them, they succumbed to death. Actually. So all these 6, they were all above 55, and this doesn't reflect the whole population that all the 55 age people will be affected, but these people are more prone to the disease, and the disease prognosis would be very high in these people. And what we saw in consistent is that fever is the common symptom out of all, and cough, 1 by 4 didn't have any symptom of cough or anything, and most fatigue or myalgia were not experienced by almost half of the population that we studied. So this is a sample study that correlates with whatever diagnostic test that we designed, and this is a very powerful implication where all the diagnostic test has to be correlated clinically. And this is the one piece of puzzle that is missing from most of the laboratories, and we did this.
Hello, sir.
Yes.
Sorry to interrupt. We have another speaker from the US, and it's already 1 a.m. for him. So we have.
It's my last slide. It's my last slide.
Yeah, we have few questions as well. Yes, thank you, sir.
Yeah. Okay. To conclude my talk, actually, I thought of presenting my NGS data. We found a very good, interesting NGS data, but unfortunately, I could not present it right now because I'm out of station. So the first thing that we found was that the testing is a main key because everybody knew it, and choosing the right primers and the chemistry, along with the quality of swabs and the spot of the swab to eliminate false negatives is the key for any diagnostic test. And I would highly recommend to follow clinically whatever diagnostic test that's been designed or validated in order to have a concrete or a full-scale positive result, because it's going to have a lot of implications in a patient or a human being's life. So I would suggest all the diagnostic tests or labs kind of go for this kind of, and also do make some amendments according to whatever chemistry that you have designed to make sure that this applies, this correlates or in synchrony with the clinical samples. And one more thing that we found is that there are some sequences that are submitted to public databases where we find some kind of an inconsistency, but despite that, most of the sequences are defined, and we do see a lot of mutations in the spike protein area, because this is the area that we are concentrating on. And I think our data would be very useful for people who are designing vaccines in the future, consistent vaccines or sustainable vaccines as of today. So thank you very much, and I would would urge everybody to be safe and stay healthy. Thank you.
Thank you. Thank you very much, sir. We have a few questions. Um, so the first question is, do you do RNA library prep for COVID-19 genome sequencing?
Genome sequence, yes. RNA library prep, yes. Yes, we do. We do library preparation. Yes. That's the first step that we do after the, and also we have an in-house so normally people in the US, they're using a lot of kingfishers nowadays to make the sample costs spiking up. So we have designed our own methodology to, uh, to find a right extraction method as well as to find the right libraries.
And what are all the potential, like the genes that are like upregulated from the genome sequencing that is like predominantly upregulated, like in your RNA-seq, for example?
Okay, so upregulated. I'm sorry, we don't look at the upregulation here, ma'am, per se. What we look at is the sequence change or a mutation, mainly with respect to spike protein. So if ever there are any changes to the submitted sequences, for example, when you go to NCBI, you see the Wuhan submitted sequence, and if this is our reference sequence that we did, and we don't look at the content of RNA actually, because I don't think it would be applying for the cells that are being infected by the virus. So as you can see that the reproducibility of index for COVID-19 is 4.5 almost. Means 4.5 crore viruses will be coming out of one infection, one spot out of epithelial permanent cells. So what we look at is the changes in the sequence, mutations or the dynamic mutations and the strain capacity.
And how long would it take to like submit those sequences and do the cross checking of any sequence changes or mutational changes that you normally see?
Basically, we have to publish this data.
Yeah.
And we want to make sure that whatever we saw is in line with the in silico model. So we saw two mutations in spike protein, which we think that makes a lot of difference in terms of affinity to the ACE2 inhibitors. Sorry, ACE2 receptors. So this is what we are modeling right now, ma'am. But this is a very academic project actually. Myself being an academic, I'm more interested in this, but we are also into clinical trials. So that's the reason why we have to conduct this test.
Yes.
NGS is not part of our diagnostic ambits. So this is out of our pure interest as well as to make sure that whatever we are designing is in line with the clinical setup. And submission wise, as soon as we publish, you'll find our sequence in the public database. I'll send you the link if you want. I'll share it to Dr. Venkatesh.
Yes, sir, would be nice. Yes, thank you.
Yes,
And
and it makes a lot of. Go ahead. Sorry.
Yeah, this is another question. Like, what is a biomarker in the diagnosis of a COVID-19, or the immunochromatography of the COVID-19 contains which biomarker? Which biomarker do you use for your immunochromatography?
Okay, that's a good question, but I'm not into immunochromatography. I'm purely molecular clinical stuff. I think I'm not the right person to answer this question, but yeah, I think it's mostly I did not get what biomarker you're looking for. You mean on the virus side or on the host side?
I think it's no. I'm not very sure about it because this is the question that came from the audience. The question.
The biomarker. No. I'm thinking like biomarker is like the one that is expressed in the blood as soon as the virus is infected, or what kind of like? I did not get what. Are you looking for what kind of protein you detect while you're doing a immunochromatography?
Yeah,
This is
I'm
what I am stuck at. So,
okay. So I think that's not something which covers into this session, I guess. And
yeah,
probably
we are not into
Patel.
biomarkers, ma'am. We are into diagnostics and how this whole diagnostic setup is going towards vaccine development and what kind of strains that are being discovered in real patients. And whatever data that I showed you right now is real data from the patients. And this is out of our own pockets and whatever money that we have invested. And
Yeah, purely using genome sequencing for diagnostics.
yeah, it's a PCR. We have also developed lamp-based technique as well, madam,
Mm-hmm.
which works very well in India basically because it doesn't need
Mm-hmm.
a lot of equipment. Basically, it needs two heat blocks and a nice program to design lamp primers. And this works very well, but it cannot be multiplexed. Right? So it's called the loop amplified primers. It's a lamp-based technique, and there's one company in San Francisco. They have already got the EUA approval for marketing it. I think this is the best thing that any academic entity could design to
And that's
detect
much better
COVID-19.
than the RT-PCR.
Uh, for it's almost equally sensitive, but it's comparatively far less expensive compared to probe
RT-PCR.
based. Yeah, because ICMR, I was reading through ICMR guidelines, and I was in touch with Bangalore Gene. So
Mm-hmm.
I was surprised that they were looking for a probe-based test with two gene compulsory on the RT-PCR, RT-qPCR, for example. So that means you have to use three probes because RNase-P would be your internal extraction control.
Mm-hmm.
RNase-P being one copy for each cell makes a very good internal extraction control. So
Mm-hmm.
that is the golden curves that you can see on my PCR curves.
Mm-hmm. I think it's
So
less expensive and even quicker because
absolutely,
you can see it on your eye basically. Because for
yes,
RT-PCR, definitely we have to
yeah,
wait for the results, and it takes some time for the preparation as well as to run the PCR as
yes.
such. So I can understand that. And before closing this session, one last question: does mutation in virus genome greatly affect the reliability of COVID-19 detection using RT-PCR?
Yeah, that yes. If your primers fall in that area, and then the mutation is not point, and it's a big one. Definitely
Yeah,
affect the overall melting temperature and so on and so forth, as you can see.
definitely. And
And it's like a lottery, I would say. Your
I think it's
primers.
more important to design primers that's in the less variable region.
Yeah, I really appreciate the move from ICMR basically because they have made a mandate that two genes have to be introduced. And right now we are using two genes now in our lab, which is RDRP as well as N1 gene, which is both a combination of WHO and CDC. And I
Yes,
would
sir.
suggest everybody who is into diagnostics would definitely go for a two gene diagnostic rather than one gene because it would be giving more false positives as per my experience is
Okay.
concerned.
And how do you deal with the false negatives, if at all there is?
That's a very good question. That's a very good question. False negatives, yes. Um, let me show you one slide actually, but as I'm limiting on time, you know, the main problem comes when a patient is falling at a very later cycle that is 35, 33 to 30, 40 cycle period. This is
Mm-hmm.
where I call this as a dangerous window because if ever you see a patient with an amplification of with RFU around 1600s and still this
Mm-hmm.
guy doesn't have any symptoms, which means I would say definitely, you know, I would make him a false positive. I would call him a
Mm-hmm.
positive basically, but he's false positive basically. So it's better
Okay.
to have a false positive than to have a false negative because false negatives are super spreaders. So
But
there is no way that we could contain.
do asymptomatic patients likely fall under the category?
Asymptomatic, no, ma'am, because the asymptomatic patients do have viral load.
Oh yes, yeah, technically right. Okay, I've been stupid.
They do have
Yeah,
a, no,
definitely
no, no, it's fine.
when they
No, no, you're
actually
fine, madam. You're fine. You're fine. I do
when
understand
they
your question.
definitely look into the swabs, and when they are doing when they are analyzing at genome level, definitely they will be detected. So there's nothing to do. I can understand there's nothing to do with the asymptomatic stuff,
Absolutely.
asymptomatic behavior of the patients. I can understand at genome level, you can clearly differentiate, the positive from the negatives.
Yes, absolutely. And
Yes,
also
I can understand that.
the asymptomatic patients are so like because they see the result as positive, but still they don't take any curative measures. That is the one of the biggest hurdles that we are seeing a lot of positives in India. So they think, okay, I'm asymptomatic. This test might be wrong actually, so, but it's not the case. End of the day, maybe his window period is a bit extended, maybe on the 10th or 15th day. So
Mhm.
this is what we saw from our studies. It's a very interesting study. This as COVID has given us, and it's a real boon for molecular diagnostics in terms of
Yes, sir.
NGS as well as introducing the different kinds of PCR into the market.